Yes, so I'm Adam Vanbergen. I coordinate the VOODOO project, and you can see the project was made of a bunch of institutions across 4 countries in Europe. So we've heard a lot about IPBES today. In 2016, the IPBES published its pollination assessment at a global scale and identified many pressures on pollinators. And in the VOODOO project, we took inspiration from that summary report, where they identified, in fact, a knowledge gap concerning the interplay between land use and disease risk for wild pollinators in particular. And so in VOODOO, we're interested in this process, and we decided to work at the landscape scale, replicated in the 4 countries I just spoke of. We examined the community organization and disease risk along gradients of land urbanization and also the intensification of agriculture. We decided in the end to focus in on specific viral pathogens of honeybees, the black queen cell virus, and the deformed wing virus. So we've known for a long time, really, that honeybee colonies, managed honeybee colonies, are challenged by a host of different viral species. And in the case of deformed wing virus, we also know that infection with the parasitic varroa mite can exacerbate the virulence of that particular virus. And we also know that when infected honeybees move into the environment looking for pollen and nectar sources, they can contact wild pollinator species at the site of flowers. And there is some published evidence at the beginning of this project, indeed, from other researchers, that wild bumblebee species have been seen to catch these viral pathogens. Next click, please. But what VOODOO was seeking to do was to assess this level of spillover of pathogens from the managed bees to the wild pollinator community. That's to say, the network of multiple species of wild pollinator species and managed honeybees in the environment. Next click, please. And importantly, what we were seeking to do was to see how the way we manage the landscape, how that affects the diversity, the abundance, and the availability of different floral resources, and what that means in terms of reorganizing and rewiring this network of community interactions, and whether that affects the probability of pathogen spillover between species. Next, please. So our first key message from the main study that we've done is really that the prevalence of viruses varies amongst the different pollinators we examined, and it is also shaped by the landscape. So if you notice at the top, the honeybee is quite clearly the primary reservoir species for both viruses indeed, and this is also confirmed in laboratory experiments. But you can also see that other pollinator groups have levels of viral prevalence in them as well. The other thing I want you to take from this slide is that as you move along the landscape gradient from an intensive agricultural landscape through to the urbanized land cover, what you see is different responses to that gradient in terms of the degree of prevalence of viral pathogens. So in fact, the situation is worsened as you move into the urban environment for the managed honeybee, but it declines for the wild pollinator groups to variable degrees. Next, please. The second key message is that the transmission of these viruses between honeybees and wild pollinators is driven by viral density and the degree of ecological niche overlap. So what we have found is that where you've got the situation of a greater density of honeybees, individuals and colonies, and that's coupled with a higher prevalence and load of viruses in those colonies, and then you add that to situations in the landscape where there's a greater degree of overlap in flora use between the managed honeybee, for example, and this bumblebee species in the purple circle there. When you have these two factors combining in the landscape, next click, please, what you end up with is a much greater risk of viral spillover from managed honeybees to wild pollinators. Next, please. So our take-home messages really today are that honeybees are confirmed as being the main viral host for both the viruses that we examined. But the virus is detected in wild pollinator groups and species, albeit in variable ways, depending on the group in question. And in terms of what we can do about this problem, Simple thing is to boost the diversity of pollen and nectar sources in the landscape. That is to increase the habitat heterogeneity of landscapes. And this will do two things. It should dilute the risk from pathogen spillover from the managed population of honeybees to wild pollinator species. And it will also directly boost wild pollinator populations, as you've just seen, in terms of their nutritional health and ultimately their diversity. Next slide, please. So I'll just conclude to thank all the members of the VOODOO Consortium, in particular all the students and the postdoc and the master students, technicians without whom the project isn't possible. And again, the funders, and just to point you to the VOODOO project website up there on the top right, where you can follow the project and where we publish all our papers and lay summaries of each paper as well. Thank you.